students, welcome back to a new video of Sunanda's tutorial. Today we will discuss about the lipid metabolism. It is the part 1 video of lipid metabolism. In this video we will discuss about the triglyceride synthesis. At first I will discuss very briefly about lipids and then I will discuss about the synthesis of triglyceride. So let's start. At first, salient features of lipids. Chemical composition. Basically the lipids are made up of carbon, hydrogen and oxygen like carbohydrates, but they are poorer in oxygen content as compared to carbohydrates. Carbohydrates have more oxygen. Chemically, they are largely esters of fatty acid and alcohol or their derivatives. So, lipids are the esters of fatty acid and alcohol or their derivatives. Then solubility. They are insoluble in water but soluble in organic solvents like chloroform, ether, benzene, acetone, etc. And thus they are hydrophobic substance because it is insoluble in water. Then polymerization, they do not polymerize to form macromolecules. They are widely distributed in plant as well as in animals. Animals are actually rich in lipids than plants. Function, they are well recognized for their threefold function like food reserve, energy source and component of cell membrane. Lipid is a component of cell membrane and they play important role for providing energy source and act as a food reserve. They also form pigment and hormones. Then types. Lipids are heterogeneous groups of organic compounds. They are primarily of 8 types such as fat, oil, wax, glycolipid, phospholipid, steroids, cholesterol and terpenes. Then the main definition of lipid. The lipid term was first coined by Bloor in 1943. He defined lipid as naturally occurring organic compound which are insoluble in water but soluble in organic solvents and yield fatty acids on hydrolysis. Bloor classified lipid into 3 broad categories. One is simple lipid. These are simple esters of fatty acid and alcohols, and then compound or conjugated lipids. Chemically they contain fatty acid and alcohol with some other chemical group. For example, phospholipid, glycolipid. Phospholipid have a phosphate group, glycolipid, then lipoproteins. These lipids form structural components of biomembrane and other various tissues. Hence, they are often categorized under structural lipids. Then the last one is derived lipid. These are derived or derivatives of simple and compound lipids. For example, steroids and terpenes. They mainly form hormone and pigment. Cholesterols are structural lipids of animal. So, among these lipids, among all these types of lipids, fats and oils, that is triglycerides or triglycerols, are the most common forms of lipid. Then let's discuss about the triglycerides. Fat and oils are called triglycerides as they are esters of one molecule of glycerol and 3 fatty acids. So in triglycerides, one molecule of glycerol with 3 fatty acids are present. They are also called triglycerols where acyl refers to fatty acid component. In animal, especially in adipose and fat tissues, they contain mostly saturated fatty acid which are solid at room temperature and identified as fats. In plants, particularly in germinating seeds and fruits, triglycerides are rich in polyunsaturated fatty acid, that is PUFA, which are liquid at room temperature and occur as soil. So in case of animals, they are present in adipose and fat tissue and contain the saturated fatty acid, but in case of plants they are present in germinating seed and fruits, and mostly they are polyunsaturated fatty acid. In case of animals, they are solid at room temperature and identified as fats, but in case of plants they are liquid at room temperature and known as oil. Chemically, triglycerides have two components. One is glycerol and the second one is fatty acid. In triglyceride, they are linked by ester bonds which are formed between three hydroxyl groups of glycerol and carboxylic acid group of three fatty acids with removal of water. So, it is one glycerol molecule, it is three molecule of fatty acids. So, in triglycerides, they are linked. In triglyceride, they are linked by ester bonds which are formed between, these are ester bonds which are formed between hydroxyl groups of glycerol and carboxylic group of three fatty acid. So, between this hydroxyl and carboxyl group, the bond is formed. And ultimately a triglyceride was released and with three water molecule. Glycerol plus fatty acid ultimately gives rise to glyceride, removing three molecules of water. And a ester bond is formed between the hydroxyl and carboxyl group of glycerol and fatty acid. Then lipid metabolism. In lipid metabolism is the synthesis and degradation of lipids inside the cell. It involves the breakdown and storage of fat, synthesis of lipids and function of lipids such as those involved in construction of cell membranes. In animals, these fats are obtained from food or synthesized by liver by the process of lipogenesis. In both plant and animals, lipids are stored in large amount as neutral, highly insoluble triacyl glycerol. They can be rapidly mobilized and degraded to meet cells' demand for energy. That means in plant as well as in animal, these lipids or triglycerides stored as reserve food. When plant or animal require energy, these lipids mobilized and degraded to provide energy to the plant and animal cell. All animal and plant have the ability to synthesize triglycerol or triglycerides. In animals, many cell type and organ have the ability to synthesize triglycerol, but liver and intestine are the most active organ to produce lipids. Then biosynthesis of triglycerides or triglycerols. Biosynthesis involves three main stages. One is synthesis of glycerol, which is the first component of triglyceride. Second one is synthesis of fatty acid, which is the second component of triglyceride. Then the main triglyceride synthesis. At first, synthesis of glycerol, it consists of two-step process. The step one is the reduction of dihydroxyacetone phosphate by NADH to alpha glycerol 3-phosphate in presence of the enzyme glycerol 3-phosphate dehydrogenase. So, in the presence of enzyme glycerol 3-phosphate dehydrogenase, dihydroxyacetone phosphate will be converted into alpha glycerol 3-phosphate. Then step 2, here the hydrolytic dephosphorylation of alpha glycerol 3-phosphate occur into glycerol in presence of the enzyme phosphatase. So this step ultimately produces the glycerol. Then phase 1. Then the phase 1 of fatty acid synthesis. At first, the initiation of chain occur. Initiation of chain is concerned with the production of malonyl coenzyme A from acetyl coenzyme A. Acetyl coenzyme A and malonyl coenzyme A is considered as two primer molecules of fatty acid synthesis. Formation of malonyl coenzyme A from acetyl coenzyme A takes place in two-step catalyzed reaction, and which is catalyzed by enzyme acetyl coenzyme A carboxylase having biotin as prosthetic group. So, the formation of malonyl coenzyme A from acetyl coenzyme A occur, which takes place in two-step process catalyzed by enzyme acetyl coenzyme A carboxylase having biotin as prosthetic group. So, it is an enzyme complex which aided the reaction, that conversion of acetyl coenzyme A to malonyl coenzyme A. Enzyme complex helps this process, and that complex consists of one protein and two enzymes. The protein is known as biotin carboxyl carrier protein or BCCP, and biotin carboxylase, that is BC, and trans carboxylase. So these three compound, one protein and two enzymes, helps in the process of converting acetyl coenzyme A to malonyl coenzyme A. Then phase 2 of the reaction, which is elongation of chain. Once malonyl coenzyme A is formed, and in phase 1, ultimately the malonyl coenzyme A is formed. And once malonyl coenzyme A is formed, then both acetyl and malonyl coenzyme A contribute together to form the long chain of fatty acid. The process requires fatty acid synthase system, or known as FAS, which includes 6 enzymes and one non-enzymatic acidic protein called acyl carrier protein. During the process, acyl carrier protein remains covalently bound to growing acyl intermediates with sulfahydryl group. The conjugated structure is called acyl SCP. Acyl SCP, that is acyl and acyl carrier proteins, and both of these are bound with covalent bond. Then FAS system, FAS means fatty acid synthase system, is located in chloroplast of the plant and cytosol of other organism. It has been mentioned earlier that FAS system involves 7 cyclic reactions. FAS cycle occur by 6 successive steps leading to the formation of butyryl acyl carrier protein. So then what are the features of subsequent cycles? In first cycle, the butyryl, the final after 6 reaction, butyryl SCP will be formed. Then the formation of butyryl SCP indicates the completion of first cycle. Then butyryl SCP leads to the synthesis of palmitic acid. So in the first cycle, only the butyryl SCP will be formed, but it is a 7-step reaction or 7-step cycle, and ultimately forming the palmitic acid. And the palmitic acid have 16 carbon. Palmitoyl SCP becomes the end product after 7 complete cycle, and then it is utilized for the synthesis of the other long chain fatty acid. Then termination of the chain. Once the fatty acid chain obtains the desired size of carbon atom, acyl group is cleaved from SCP. Acyl group is cleaved from acyl carrier proteins. Hydrolytic removal of palmitoyl group from SCP results in the release of palmitic acid by the action of thioesterase enzyme. So you have to remember these names of the enzymes because it is very, very important. Then the overall reaction. What is the overall reaction of this fatty acid palmitate or palmitic acid? At first the 8 acetyl coenzyme A in the presence of 14 NADPH and 14 proton with 7 ATP ultimately gives rise to palmitate with 8 coenzyme A removed and 14 NADP plus 7 ADP and 7 inorganic phosphate with the removal of 6 water molecules. So it is the overall cycle of fatty acid synthesis. So at the end of this, we have glycerol which is prepared, then we have fatty acid prepared. Then now comes the biosynthesis of triglycerols or triglycerides. So the overall synthesis of triglycerides have 3 steps. At first, glycerols should be prepared, then the fatty acids should be prepared, then the triglyceride synthesis. So synthesis of triglycerides have 3 main pathways. One is dihydroxyacetone pathway, one is monoglycerol pathway, and the next one is SN-glycerol or Kennedy pathway. At first, we will study about the Kennedy pathway or SN-glycerol pathway. The main biosynthetic route to triglycerol is SN-glycerol pathway or Kennedy pathway, as it is first described by Professor Eugene Kennedy and colleagues in 1950, by means of which more than 90% of liver triglycerols are produced. So it is the overall pathway. From glycerol 3-phosphate with 1 fatty acyl coenzyme A, lysophosphatidic acids are formed. So, at first, the glycerol 3-phosphate attach with the fatty acyl coenzyme A, and in presence of glycerol acyl transferase and releasing 1 coenzyme A, ultimately the lysophosphatidic acid is formed. Then another fatty acyl coenzyme A is added and ultimately producing phosphatidic acid. Then diacyl glycerol is formed. Then from triacyl glycerol, another 1 fatty acyl coenzyme A is added. In the presence of diacyl glycerol acyl transferase, triacyl glycerol is formed. So it is the overall pathway of overall SN-glycerol pathway or Kennedy pathway releasing triacyl glycerol. Then the dihydroxyacetone phosphate pathway. In the second pathway for triglycerol biosynthesis, dihydroxyacetone phosphate in peroxisomes or the endoplasmic reticulum can be acylated or acyl group is added with fatty acid coenzyme A esters by specific acyl transferase to form 1 acyl dihydroxyacetone phosphate, which is reduced by dihydroxyacetone phosphate oxidoreductase to form lysophosphatidic acid and can enter the pathway of above. That means when the lysophosphatidic acid is formed, then it will follow the same pathway of SN-glycerol. So once the lysophosphatidic acid is formed, it will again enter as said in the SN-glycerol pathway. That means then the phosphatidic acid will be formed, then diglycerol, then triglycerol. So ultimately producing the glycerols. So it is the dihydroxyacetone phosphate, in which at first the precursor of triglycerol is dihydroxyacetone phosphate. Then 1 acyl dihydroxyacetone phosphate will be formed, then lysophosphatidic acid, then from lysophosphatidic acid, phosphatidic acid, then is SN-glycerol 1 to diglycerol, then ultimately triglycerol molecule will be formed. Then monoglycerol pathway, it is the third and last pathway. In the enterocytes of intestine after a meal, up to 75% of triglycerol or triglycerides are synthesized via this pathway or monoglycerol pathway. Here the monoglycerol with the presence of the enzyme acyl coenzyme A transferase ultimately form the diacyl glycerol. Then from diacyl glycerol, triacyl glycerol will be formed in the presence of the enzyme acyl coenzyme A transferase. So it is a very simple pathway which generally occur in intestine in case of animals. So these are three pathway of triglyceride synthesis. So it is the part 1 video of your lipid metabolism. In my next video, I will discuss how the breakdown of these triglycerides occur